Good afternoon. My name is Jeffrey Baum, and I am the Executive Director of Michelson Philanthropies. We could not be more proud to partner with Keystone in its 50th year to sponsor the meeting this week. And I want to extend our institutional thanks and my personal thanks to Deborah Johnson and her team at Keystone for pulling this together amid months of uncertainty, and I'm so grateful to everything they've done to make this a successful and smooth and productive meeting, as well as to everyone who made the effort to make it to Breckenridge, Colorado. Starting with Dr. Graham's keynote address yesterday, each session has been thought-provoking and energizing. It is truly inspiring to see brilliant minds come together here at this moment in human history. You are needed more than ever as we tackle these biological threats, but also social and political challenges. As I was driving here from the airport, the car in front of me had a bumper sticker. It read, quote, "The media is the virus." As one who began my career in journalism, that was a little troubling. But even more troubling was the data released yesterday and previously that shows that hundreds of thousands of Americans were lost to COVID after vaccines became widely available. We have so much work to do, both in the research labs in which you toil, but also in the communities where we live. Michelson Philanthropies was started by Dr. Gary Michelson to do just that, to promote scientific breakthroughs and to push for positive social change in a variety of areas. We have 4 foundations. They focus on medical research, education and equity, intellectual property, and animal welfare. You can read more about our foundation in the materials at your table or on the Keystone Connect app. I hope you've downloaded all of that. One key priority is why we're here today, to champion brilliant early career researchers. We support them with grant funding and public recognition to accelerate their careers while underscoring the need for greater investment in science. We are currently accepting applications for new grant funding, and there's more information at the flyers at your table. Our request to each of you is to encourage those within your networks to consider applying for these awards. Together with our partners at the journal Science and at the Human Vaccines Project, we're eager to find and work with young researchers around the globe who focus on immunology and vaccine research. On behalf of Dr. Gary Michelson and his wife, Alia, today we honor this year's Michelson Laureates. Dr. Michelson is the driving force behind this effort. He is a board-certified orthopedic surgeon and inventor who revolutionized spinal surgery by developing hundreds of new therapies and tools. After selling his portfolio of patents in 2005, he now devotes himself full-time to philanthropy. He's in Los Angeles today, so he and his wife used an iPhone, so you'll see the quality, to record a message to share with you.
Good morning. Thank you for joining us to honor these remarkable young scientists, the recipients of this year's Young Researcher Prizes. While the scope of my wife Alia's and my philanthropic interests are rather far-ranging, nothing has the potential to have a greater beneficial impact than does medical research. And inasmuch that it would seem that it is our own immune systems that are at play in almost every disease that afflicts us, obesity, and even aging itself, no field of medical research has more potential to help us than does immunology. That is why we are here today. We are all already up to speed on the story of former President Jimmy Carter, who was diagnosed with far progressed metastatic malignant melanoma, historically a rather short-fused death sentence. But he was cured when his own immune system was enabled so that it could detect and respond to the cancer cells. And for today's trivia, now the longest living president in American history. The immunological advances as they relate to oncology have been so great that the term immunotherapy has become nearly synonymous with cancer treatment. But in fact, immunotherapy is any manipulation of the immune system to either prevent or treat disease. And we have actually been doing that quite effectively and for a very long time with vaccines. Investments in science and innovation are a down payment on our future. Foremost in medical research, immunology and immunotherapy have the highest potential to transform the trajectory of human healthcare. As an aside, today in California, we are doing just that. A group of philanthropists with remarkable resumes related to medicine, science, the building out and operation of best-in-class incubators and accelerators, the birthing and financing of biotechnology startups, and much, much more, have come together to help fund and create what will be the largest and most comprehensive center for immunology and immunotherapy writ large with a capital I in the world. A leading university has made available an on-campus site in close proximity to a world-class hospital system. And California Governor Gavin Newsom has proposed a $500 million investment to help create what will be the California Institute for Immunology and Immunotherapy. I mention this now because it will not be business as usual, but just on a larger scale. First, we intend to de-silo all of the brilliant scientists across all of science to work together on project-directed research in a manner similar to the Manhattan Project. Then, we intend to encourage and finance imaginative, creative, high-risk, high-return research, and to fully fund young scientists to be able to conduct their own research rather than laboring for someone else. So in many ways, what we will be doing at that institute is what we have already been doing with our Young Researcher Prizes and Awards. In awarding these prizes with Science Magazine and the Human Vaccines Project, we identify researchers who are 35 or younger and help them to pursue their ideas that dare to push past the known limits and beyond the orthodoxy of the moment. Today, we recognize the bold imagination of these Michelson Prize winners. These prizes advance immunology research, and they have also accelerated the trajectories of our past prize winners' careers. Together with my wife, Alia, we congratulate these winners, thank them for their work, and wish them Godspeed on the fabulous journeys that lie before them. Thank you.
If you would like to learn more about our grant programs or the new California Institute for Immunology and Immunotherapy, please see me or my colleague Michelle Morris, who's standing, and you've seen her at the table during the conference, and we'd love to speak with you. But now for the prize winners here with us today. After a brief video introduction, they will make a short presentation and welcome your questions. First, we have Dr. Rong Ma. She is the winner of the Michelson Next Generation Grant presented in partnership with the Human Vaccines Project. Dr. Ma.
My name is Rong Ma. I'm a postdoctoral fellow at Emory University. I received the 2021 Michelson Prize for harnessing receptor mechanics as a marker for immunogenicity to isolate and identify potent T-cell receptors and recognized antigens. Curiosity has always been one of the biggest motivations that pushes me forward in research. My research focused on developing molecular sensors made of DNA, which can detect receptor forces on piconewton scale generated by cells when they are probing the surrounding environment. We and others in the field have found that the T-cell receptor employs mechanical forces as a part of its triggering mechanism in order to boost this specificity of antigen recognition. And by studying these forces, we found that the mechanical sampling of the antigen by the T cells is correlated with the T cell activation. So with this knowledge, moving forward, we are trying to develop an assay to harness these mechanical forces transmitted through the receptors as a biomarker. This would be one of the very first attempts of harnessing the dynamic and biophysical nature of this antigen recognition process for potential cancer immunotherapy purposes. And in the long run, if this method is demonstrated to be effective for precisely identifying the potent T-cell receptor antigen bindings, we could use it as a strategy to generate personalized neoantigen cancer vaccine. As an early career researcher, the support from Michelson Prize will give me a lot of independence, as well as the peace of mind to focus solely on this research. And it allows me to pursue the high-risk, high-reward project of developing a novel assay for neoantigen and neoantigen-specific T-cell identification.
Hello everyone, thanks for this opportunity. I'm very excited to share some of my research today, and the title of my talk is Mechanical Forces Help T-cell Receptor Distinguish Friend from Foe. So T-cells constantly scan the surface of other cells to defend against infections and cancer. As shown in this video, a cytotoxic T-cell is scanning the surface of an antigen-presenting cell. And if we zoom in at this interface, we will find one critical molecular interaction, which is the T-cell receptor binding to the peptide MHC molecule. And once the T-cell receptor recognizes the peptide, it initiates the signaling cascade. And as an outcome, the T-cells get activated and produce these cytotoxic granules and precisely release them to the target cell to kill. Now this process occurs at the interface. So we were wondering, is there any physical cues that the T-cells harness in the recognition and the activation? For example, is there any traction forces that the T-cells generate when they scan for antigens on the surface of the target cell? So one particular question that we are asking is that are mechanical forces involved in the TCR peptide MHC interactions, and does this force matter for the T-cell activation? To answer this question, we needed to engineer substrates that are chemically identical but mechanically different. So we came up with this strategy of using tension gauge tether technology. So this tension gauge tether is a DNA duplex that is anchored on a glass cover slip and presents the peptide MHC ligand for the T-cells. And these DNA duplex would denature under different forces. And based on this positioning of the peptide MHC ligand, it can either denature in the unzipping geometry at 12 piconewtons or the shearing geometry at the 56 piconewtons. And just to give you a sense of how, on the scale of these forces, 1 Newton is how you feel like when you hold an apple, and a piconewton is a trillionth of that. So once these tension gauge tethers are denatured, the mechanical transduction from the TCR to the peptide MHC is terminated. And from these microscopy images that I'm showing here, as you can see, if the mechanical transduction between the TCR and the peptide MHC is terminated at 12 piconewtons, we see a significantly decreased level of phosphorylated ZAP70, which is a marker for T-cell activation. So this data tells us that sustained mechanical forces through the TCR is very important in T-cell activation. And now the question for us becomes, can we measure these forces? The way that we measure these forces is our lab developed this DNA-based molecular tension probes. The way that this probe works is we anchor these probes on a glass cover slip and present the peptide MHC molecule on top of it. When there's no force, this hairpin module is folded, so there is no fluorescence from this Cy3b molecule because it's quenched by the BHQ2 quencher here. As the TCR comes in and binds to the peptide MHC molecule here, if it pulls on this hairpin module and exert force that is greater than 4.7 piconewton, this hairpin will denature, unfold, and then separate the fluorophore quencher pair, and thus we see a fluorescent signal. So with this method, we were able to visualize the TCR peptide MHC forces in real time. And what you're looking at is a time lapse of these forces. Each of those fluctuating green dots under the footprint of the T cell represent the forces that are greater than 4.7 piconewtons exerted through individual TCR peptide MHC interactions. And by using the DNA hairpins that unfold at different force levels, we were able to find a very defined range of these TCR forces, which is between 4.7 piconewton to 12 piconewton against the antigens during T cell activation. So now we know the magnitude of the forces. We're very curious whether the overall cumulative TCR forces would be different if we present peptide ligands with different potency. The way we studied this is we included a complementary strand to the hairpin stem loop part. This complementary strand would only bind to the mechanically unfolded hairpins, thus locking this fluorescence signal so that we can do a quantitative measurement. And as you can see from the microscopy images here, for the OT1 CD8 positive T cells, N4 here is the cognate peptide. It generated the strongest force signals. And the Q4, V4, and G4 are the altered peptide ligands with less potency. And these peptide ligands with a single amino acid mutant at the fourth position had significantly decreased force levels compared to the cognate antigen. And these peptides also share a similar level of affinity to the TCR, so which really outlines how important we use mechanics as a marker for immunogenicity because it's highly correlated despite the similar level of affinity. Moving on, we were also very interested in the kinetics of these TCR peptide MHC forces, and then we were interested in measuring the force lifetime. The way we studied this is we further modified our method, so we extract the lifetime information from our tension measurements. And as you can see from this figure, the stimulatory antibody, anti-CD3, has the longest force lifetime above 4.7 piconewtons for the TCR CD3 complex, and the cognate peptide N4 has over 10-fold longer lifetime comparing to a less potent Q4 antigen. So this result also echoes with some other research in the field by the single molecule force spectroscopy. The way that these researchers study these forces is they coat a bead with peptide MHC molecule and apply force through individual TCR to the T cell. And then with this method, they were able to identify some TCR peptide MHC interactions that forms catch bond, meaning the lifetime of this binding peaks at around 10 piconewtons. And then most of the peptides are forming slip bonds with these TCRs, which means the lifetime of the binding decrease as the force is applied. And a lot of these research in mouse models suggests that catch bond is a key feature for strong TCR signaling. And recently, Garcia Lab at Stanford also identified 2 human TCRs that bind to the same antigen with the same level. Here's the tetramer staining. However, only one was able to activate the T cell and initiate the immune response. And from the single molecule force spectroscopy characterizations, the only difference there is this activating TCR peptide MHC interaction was able to form a catch bond, and the other one is a slip bond. So with that, I'd like to conclude my talk. What we find is the T cells generate 4.7 to 19 piconewton tension through TCR against the cognate peptide MHC, and terminating this force at 12 piconewtons would dampen the TCR activation, the T cell activation. And this TCR peptide MHC force is correlated with the potency. And furthermore, we find that the force lifetime at 10 piconewton is a key difference between activation or no activation for the T cell. And we are hoping that our research on the TCR mechanics with others in the field would inspire people to integrate this component of the TCR peptide MHC interaction in developing immunotherapies and also vaccines in future. And at last, I'd like to thank my advisor, Khalid, and the Salida Lab as well as the collaborating labs and also the funding support from NIH and the recent support from Michelson Foundation and also Human Vaccine Project. Thank you.
Yeah, that's a beautiful system. I was just curious about what's the impact of the TCR docking angle on the ability to form the catch bonds?
Actually, that's a very good question. Actually, we don't really know the docking angle because to really know the angle, I think you will have to have some structural studies. And to do a structural study on a membrane protein is already hard enough. Not to mention to have it under the strain, because you have to look at the crystal structure under the strain, which is really hard. But from some modeling support, data support, I think people are discussing the possibility of the FG loop on the TCR that would bend and press on the CD3 when there's mechanical forces, and that could be one reason. And there's also studies suggest that as this force is applied, not just TCR goes through this conformational change and press on the CD3, but also the MHC has a conformational change that would facilitate this sustained force and elongated lifetime under force for the binding.
Thank you very much, Dr. Ma. Next we have Dr. Camila Consiglio of Keralinska Institut. She is also a winner of the $150,000 Michelson Next Generation Research Grant. We're pleased to show a video and then welcome Dr. Camila Consiglio.
I'm Camila Consiglio, and I'm a postdoctoral researcher at Keralinska Institut. And I received the 2021 Michelson Prizes to understand how testosterone impacts the human immune system. I chose to become a scientist because I've always been very curious to understand the world around me. And so I chose specifically to go into the biomedical field to try to understand why people develop diseases and to try to find ways to better treat these diseases and prevent them. So my research is trying to understand how the immune system is different between men and women. And so we know that, for example, men tend to have more severe infections when compared to females. But on the flip side, women tend to have higher responses to vaccinations. So why is this? Why is it that women and men have these differences in immunity that can contribute to their susceptibility to different diseases, such as infectious diseases and autoimmunity? My research is trying to understand how a specific sex hormone, testosterone, that is different between males and females is impacting the immune system of humans. And so by trying to understand the impacts of testosterone in immunity, we can better understand how to prevent and treat infectious diseases and design optimal vaccine strategies that can fit one's biological sex. And so in my research, I'm combining highly advanced technologies that allow you to look at different components of the immune system all at the same time with computational methods. The Michelson Prizes will help me address a question that I've been interested in for a really long time, of how is it that males and females have differences in their immune responses. And so by understanding how biological sex impacts the immune function, we can now understand better how diseases are developing differently between males and females, and also optimize strategies to treat these infectious diseases, such as developing vaccine strategies that are fitting to one's biological sex.
Hi everyone, I'm Camila, as you just saw me in this video. I'm really pleased to be here today, and I thank the Michelson Group for this opportunity. And I'd like to tell you a little bit about this work that I've been doing, understanding sex differences in immunity. And so a little bit of the background here is one question that I'm really interested in understanding is how does immune variation contribute to disease in humans? And let's take COVID-19 as an example here. We know that people infected with SARS-CoV-2 can develop a variety of different symptoms from no symptoms at all the way to severe disease and death. And although we have now identified a few factors that can predispose to severe disease, we still don't know who will develop which. Therefore, it's highly important that we try to understand how the immune system differs between individuals so we can not only better prevent and treat infectious diseases, but also other immune-mediated diseases such as autoimmunity, cancer, and so on. And so what are the factors that can mediate these differences in humans? One factor that I'm really interested in is biological sex. And this is because biological sex impacts immune function and disease susceptibility. So if you look over here, and this is something we've come across recently with SARS-CoV-2, we know that men tend to have higher hospitalizations and deaths when compared to women, but this is not strictly just shown in infections with SARS-CoV-2. In fact, infections tend to be more severe in men when compared to women, and this is shared across different types of infections: viral infections, bacterial, fungal, and parasitic. Also on this side, we know that men also develop cancer at a higher rate when compared to women and also have more deaths associated with cancer. And on the flip side here, we know that vaccine responses tend to be higher in women. So if you look at IgG levels generated post-vaccination, women tend to have twice as high levels when compared to men. So you can conclude by this that women are great and we're doing the right thing, but this is not really the case because it comes with this adverse side effect of this maybe heightened immune response, right? You also have more vaccine side effects in women, and women will also develop more autoimmunity when compared to men at almost a 90% to 10% rate. And so why is this? Why is it that we see these differences between males and females? And is biological sex maybe an immune thermostat that could be turned up and down? So what are these reasons, and how are we trying to understand them? There are three major factors that underlie these sex differences in immune responses and disease susceptibility. One of them is societal factors, behaviors that are different between the sexes. We also know that within genetics, for example, in the sex chromosomes, there are immune genes that are expressed in the X chromosome, and they're highly expressed in females when compared to males. So there are some differences there. But there's also sex hormones that are different between males and females and vary throughout their lives. And so now, what do we do in this field? How do we try to tease this apart? And we have a few challenges. The first one is that it's really hard in humans to dissect the genetic from the sex hormone effects because they kind of overlap, right? And the second difficulty here, the second challenge, is that we really need to have a holistic understanding of how the immune system functions to really try to put together how is a B cell functioning in males, a T cell, and so on. And so we know from studies where we take blood samples that you have a variety of different immune cells that are interacting with one another. You have protein, peptides, components, cell types, functional states. And so how is this interplay, right? And so classic immunology studies tend to be very mechanistic. They'll look at one cell type at a time and really go deep into mechanistic understanding. But we're really being more low throughput. And so the idea here is that in our studies, we're trying to go beyond this and have a more of a holistic understanding of how the immune system is differing between males and females. And so putting this together, how are we teasing apart the sex hormone and the sex chromosome effects? We are looking at subjects that are undergoing sex reassignment therapy. So females that will come into the clinic, as you know, they'll have a fixed genetic background of XX chromosome, and they'll start transitioning from a female to a male sex hormone background. So they'll receive sex hormone treatment, they'll receive testosterone. And when we look at their immune system, we do this by doing blood collection before and after transitioning. We can try to infer and understand how is it this testosterone treatment is impacting their immunity. And so we do this by looking at different components of the immune system. We look at the plasma proteome, the immune cell phenotype and composition, and the transcriptome as well. And in a nutshell, we have found that the testosterone treatment in these subjects that are undergoing a female to male transition is changing significantly. So this is just a brief overview. You can see that they'll receive testosterone treatment and their testosterone levels go up to a male range, whereas their estradiol levels remain the same. And what is this implicated in? We can see through bulk transcriptomics and the plasma proteome that there is an induction of inflammatory state when these subjects are receiving testosterone. We also see phenotypic changes in plasmacytoid dendritic cells, and also their composition is decreasing over the course of testosterone treatment. And ultimately, we also see changes in B cell function. And so if you're interested in knowing a little bit more about these findings, please talk to me at the poster session right after this lunch. And ultimately, I just want to go a step beyond here. So these are our findings. We are seeing that testosterone does significantly impact the human immune system, but how is this happening? And with the Michelson Prize, I'm able to address these next questions, where I'm looking at both functional responses and more mechanistic studies. And so in terms of function, what I've shown to you briefly today is that at baseline, these subjects are changing. But what is actually happening functionally to them? So when they're stimulated, are they able to create like better immune responses to a virus, to a bacteria? What's actually happening? So we're doing this with the Michelson Prize, and we're also looking mechanistically. So what happens with testosterone? Is it directly interacting with immune cells, and because of that, their phenotype, their functions are changing, or is this something indirect? So we're addressing these questions now, and I would really like to acknowledge the collaborators we have in Sweden. So the recruitment of these subjects have happened in different institutes in Sweden. In our lab, which we have a highly computational and experimental background to be able to really profile these subjects over time. And I'd like to also acknowledge our fundings, and especially the Michelson Prize, and I'd be happy to take any questions or discuss during the poster session. Thanks.
I'll ask a question. So it's curious, you talked about the B cell responses, but we've taken care of HIV patients for years, and one of the things we find is, especially for the people with very low CD4 counts, men and women starting at the same CD4 counts, women recover way better and men don't. Have you looked at or any interest in sort of understanding what testosterone might be doing there in terms of immune recovery as people are put on therapy?
That's a really good question. So the HIV field is within the viral infections, knowing that there's these sex differences. The viral load in HIV tends to be different between males and females, as she's saying, and the also exhaustion phenotype is observed differently between males and females. And so this is also a cohort. I know a group that works with this in Zurich, and they're looking specifically at HIV-infected subjects. And within their cohort, they have transgender subjects and cisgender subjects. So they're able to compare, the receiving of sex hormones or not. But we are not addressing this ourselves, although we're doing these functional experiments in vitro with some stimulations to see, looking at PBMCs before and after treatment, how are they changing functionally? Yeah. Thanks.
Hi, I have a quick question for you then. But have you attempted to control at all for the differences between naturally produced testosterone and synthetic testosterone provided to transitioning patients? Or have you thought of using people with abnormal genetic phenotypes that produce, different levels of both hormones?
So in terms of the testosterone that they're receiving, they all are receiving somewhat of a standard treatment. One thing to note here is this cohort can be enrolled after they're 18 years of age. So that means they have already undergone puberty for a few years, and then they enroll. So they've already, had this exposure with testosterone, and then they start, with other sex hormones, the opposite sex hormones, and then they receive testosterone. And sorry, what was the second question?
Sorry, it was also if you had thought of looking at these markers in individuals without with
Absolutely.
genotypes different than XX
Absolutely.
and XY.
So that's a really good comparison to have, because we do want to look, you see this is a longitudinal study, so we have before and after, right? This is a really good way of understanding these variations over time and comparing with cisgender individuals as well. Within the work that I'm doing, in this bigger study where we're also accounting for sex chromosomal changes, for example, and we could be able to look at other abnormalities. For example, there's some phenotypes in women, for example, PCOS, where there's higher testosterone production, and you could maybe try to pair that, but we haven't done that yet. It's a really good idea.
Awesome. Thank you.
Thank you.
Thank you so much, Dr. Consiglio. So Dr. Consiglio and Dr. Ma were recipients of the Michelson Prize Next Generation grants. Those are research grants for research proposals. And as you can see by the flyer, the application period for that is currently going on. It's a short initial application, just a letter of interest and pretty much just an abstract. And then a screening committee will review all those and then invite further applications. The other prize that is new this year is something that we started with the journal Science and the American Association for the Advancement of Science. And this is a prize for completed research that then will be recognized by publication in Science, as well as a cash prize as well. And so we're delighted today to also have a finalist for the Michelson Philanthropies and Science Prize for Immunology, Dr. Scott Bering from the University of California at Berkeley.
My name is Scott Biering, and I'm a postdoctoral scholar at the University of California, Berkeley. And I was a finalist for the Michelson Philanthropies and Science Prize for Immunology for my essay on antibodies targeting the non-structural protein 1 of flaviviruses. I got interested in the field of immunology because I came to appreciate that viral pathogens only exist in the face of coevolution with our immune systems. So I wanted to understand how our immune systems recognized viruses as invading pathogens and use that information to produce new therapeutics and vaccine strategies targeting these viral pathogens. My current research investigates how emerging viral pathogens cause severe disease in humans. So we focus on two viral families at the moment, the flaviviruses, which are mosquito transmitted, and they include dengue virus, Zika virus, and West Nile virus, and more recently SARS coronavirus 2. We specifically want to use this knowledge from our research to, one, produce new therapeutic and vaccine strategies against these current viruses, the flaviviruses and the coronaviruses, but we also want to apply general themes to even new emerging viruses that we don't know about yet. My ultimate motivation that keeps me going is really twofold. It's both a passion for alleviating global disease burden with the research that I do, but it's also mentoring a new generation of scientists. The Michelson Philanthropies and Science Prize for Immunology will have a great impact on my ongoing career, both because the funding will help me to set up new systems to understand how viral pathogens cause disease, but also to help me recruit new lab members and get an independent investigation position to continue this interesting avenue of investigation.
So as that's getting up, so as you saw, my name is Scott Biering. I'm a postdoc. I'm very excited to share some of our research with you today. I'd like to thank Michelson Philanthropies and Science for this opportunity, and Jeff and Michelle for planning this wonderful event. So here's my talk right here. So today, I'm going to tell you a little bit about our work on the Flaviviridae, how they cause disease. I'm going to talk about non-structural protein 1 of this viral family, and how this protein could be targeted as an alternative vaccine strategy to targeting the envelope protein. So the Flaviviridae are transmitted by Aedes species mosquitoes, and they're widespread in any area that house these mosquito habitats. The Flaviviridae are a very diverse family of viruses that, while they possess a very similar genome and overall viral life cycle within a cell, they cause very different disease severities. And you see them cause infection in different tissues, so their tropism differs quite a bit. And this is relevant for the protein we're going to talk about, non-structural protein 1. So dengue is considered to be something of a more systemic infection associated with vascular leak and endothelial dysfunction, something similar for yellow fever, while Zika, West Nile, and Japanese encephalitis have more of a neurotropic tropism, where you find them more in the brain. And one of the main challenges to producing, at least in the situation for dengue, a vaccine targeting the main target, the envelope protein, is the fact that there are 4 major dengue serotypes, and antibodies raised against one serotype may produce homotypic protection, but then you'll also get heterotypic antibodies raised that may be sub-neutralizing to the additional serotypes. And this can actually enhance infection by allowing these viruses to infect immune cells by being taken up in an Fc gamma receptor dependent manner. So basically this has made it challenging to produce a vaccine targeting the envelope protein. So it's been quite important to investigate how else we can therapeutically target flavivirus infection. And one approach that we took was to look at other proteins that may contribute to severe disease. And one of my favorite proteins, this protein is just wildly multifunctional and very interesting and very atypical for proteins. This is non-structural protein 1 of dengue viruses. Critical for viral replication. You find it intracellularly associated with dimers as a component of the replication complex, but it's also secreted from infected cells, actively secreted, and it has these hydrophobic domains that it sequesters away by taking up lipid cargo, and it causes this hexamerization. You see this trimer of dimers. And what you can see is that in patients infected with dengue or other flaviviruses as well, we have a nice study on yellow fever on this now, that the levels of circulating NS1 in patients correlate with disease severity. So what you can appreciate here is that patients with dengue hemorrhagic fever, or more severe form of disease, have higher NS1 levels than patients with dengue fever, which is less severe. And now this extracellular NS1 doesn't just serve as a diagnostic of infection, it also has been shown in many different assays to contribute to severe disease. And one way it does this is by activating platelets and immune cells. It inhibits complement. And what we've been particularly interested in is how it triggers endothelial dysfunction, resulting in vascular leak, this major hallmark of flavivirus infection. And this isn't specific for dengue. We've had some studies now showing that flavivirus NS1 from multiple flaviviruses causes vascular leak, but in a tissue-specific manner that mirrors the tropism of the respective flavivirus. So here I'm showing you an in vivo vascular leak assay where you can intravenously administer NS1 of a different flavivirus to a mouse, and then give it a dextran 680 tracer dye, and you can measure relative levels of leak in different tissues. So now all of these viruses, besides yellow fever virus, have some tropism for the brain, and you can see accumulation of this dextran 680 in the brain relative to control into yellow fever. In the lung, which is really only tropic for dengue, as we see a significant enhancement of leak in the lung compared to others. And now in yellow fever, in the liver, you can see yellow fever NS1 and dengue NS1 cause leak. So what I want to show you is that yes, this protein contributes to the severe disease manifestations, but it does so in a tissue-dependent manner. So we thought that maybe we could target this disease severity to treat flavivirus infection. And so the approach we took was a structural approach to basically ask how antibodies that could neutralize NS1 function bound to NS1 and how this information could be utilized to create new therapeutics. So we teamed up with Janet Smith's lab at the University of Michigan, who had originally solved the structure of dengue NS1. And so what I'm showing you here is a dimer, 2 monomers that are associated. This is the functional unit of NS1, and it has 3 unique domains: the beta role, the wing domain, and the beta ladder. And basically the beta ladder and the wing domain are believed to mediate most of the interactions with cells. So by using an anti-NS1 antibody that was protective, we could ask both which domains are protective and potentially produce new therapeutics. So we decided to use an in-house produced antibody that was actually produced in an immunology class at the University of California, Berkeley, probably 15 years ago. And we and others in the flavivirus field use this antibody as a major tool to study pathogenesis. And you can show that this antibody given alone, compared to an IgG control, is protective against a dengue virus lethal challenge model. So we thought this was a good tool to investigate the structural basis of NS1, anti-NS1 antibody protection. So again, teaming up with Janet Smith's lab at the University of Michigan, we solved the crystal structure of NS1 in complex with the 2b7 FAB. And what you can see is that this FAB binds to the beta ladder domain of dengue virus NS1, and it binds at this funny arc-like projection. You would predict this hydrophobic surface to be a domain that interacts with cells. So we predicted that this antibody is potentially very protective, one, because it may bind to critical residues in this one beta ladder right here, but then the wing domain, which is right here, which makes contact with lipids on cells, is also being sterically hindered. So we produced NS1 mutants. We had a good enough resolution where we could define the specific contact sites in the beta ladder. And as I mentioned before, this predicts that that the beta ladder is important for pathogenesis, but then also the steric hindrance model of the wing domain may also be important. So we first took a mutagenesis approach to ask which of these domains was actually important for pathogenesis. So we made a series of NS1 mutants, expressed them and purified them. And then what I'm showing you here is an in vitro endothelial dysfunction assay, so a TR assay, transendothelial electrical resistance. This measures the permeability across a monolayer of endothelial cells seated in a transwell. And with control proteins, you can see a higher level of endothelial dysfunction. And what you can see is that these three mutants that come from the 2b7 epitope of the beta ladder, they are now dysfunctional in dropping TIR. And then this other domain right here, NS1WWG, this is a critical site that was predicted to interact with the surface of endothelial cells that is now predicted to be sterically hindered by the 2b7 epitope. So by a mutagenesis approach, we could support this model. And what's interesting is that the 2b7 epitope amongst flaviviruses is actually quite conserved. You see different classes of epitopes. So the site is discontinuous, and obviously it was raised against DV2, so it's most closely predicted to bind to dengue NS1, but it's also predicted to bind to many different other flavivirus NS1 proteins. And using a capture ELISA, you can demonstrate this, that 2b7 binds very strongly to NS1 for multiple flavivirus NS1s. And then it also binds to many different flavivirus NS1s as well, in a manner consistent with how related it is to dengue virus 2. So Zika, St. Louis encephalitis, West Nile, Japanese encephalitis, and Usutu virus all bind quite strongly to this antibody, and yellow fever NS1, which is the most distantly related, doesn't bind as much. So we predicted that this cross-reactivity may actually result in protection in vitro and in vivo. So here I'm showing you the capacity of NS1 from West Nile virus or Zika virus to bind to endothelial cells in the presence of full-length or FAB of this antibody. And you can see that 2b7, which is not raised against West Nile or Zika NS1, is sufficient to block interaction with the surface of endothelial cells. And in fact, in a Zika virus lethal challenge model, 2b7 also offers protection. So basically, that's most of what I want to show you today. This is a proof of concept paper, and right now we're expanding this concept to other flavivirus NS1s, and we're making new versions of 2b7 that are more highly cross-reactive with different flaviviruses to see if targeting this epitope can be cross-protective. So what I showed you is that 2b7 is a flavivirus NS1 cross-reactive antibody that can block endothelial dysfunction and severe infection of multiple flaviviruses. Crystal structures can show that 2b7 binds to the beta ladder and also predicts steric hindrance of this other domain, the wing domain, and that 2b7 can inhibit pathogenesis from multiple flavivirus NS1 proteins. With that, I would like to acknowledge that this is a very collaborative project. So my mentor, Eva Harris, is a fantastic mentor with a lot of really willing to try crazy ideas, which I really appreciate, has allowed me to really explore a lot of things. Janet Smith at the University of Michigan, who did the crystallography and solved the original crystal structure of NS1, which has been fantastic. David Akey, the primary crystallographer at the University of Michigan, and Dr. Robert Beatty, who made the antibody originally. So today I talked to you most about this 2b7, but a lot of my work right now is investigating the host factors that NS1 interacts with on the surface of cells. And I have a poster right over there that you can come and check out later if you're interested in hearing more about that. So again, I would like to thank Michelson Philanthropies for this opportunity, and any questions you may have. Thank you very much.
Well, thank you. I also want to acknowledge the other Michelson laureates that were honored this year with the Michelson Philanthropies and Science Prize in Immunology went to Dr. Lisa Wager, of the University of California at Irvine, and then also the Michelson Philanthropies and Science Prize for Immunology grand prize went to Dr. Paul Bastard of the Imagine Institute in Paris, and his essay that was published in Science is available at your tables, and you can see that. The other honoree for the Michelson Prize Next Generation grant was Dr. Nicholas Wu of the University of Illinois. So colleagues, if you can join me in honoring this year's Michelson laureates with a round of applause, that would be great. As I mentioned, both Dr. Bering and Dr. Consiglio are displaying their research at the poster session following lunch, so be sure to stop by and visit all the poster sessions and you'll see lots of information. We sincerely thank you for joining us here today, and to those who are watching online, and we look forward to staying in touch. Enjoy the rest of your afternoon. Thank you.